DNA. This DNA how can be useful for us for analysis to identify the individual. So how with the two parts, whether it can be used as a source, how we are going to use it for identifying the basic science of DNA fingerprinting we'll be discussing in this presentation. I am Dr. Alok Dubey. I'm working as an associate professor in the College of Dentistry in Saudi Arabia.
Okay, okay, okay.
To begin with, so whenever a dead body comes to a forensic department, the first Herculean task for them is to find out whose body it is. So what conventionally what they do, they compare the antemortem and the post-mortem records. Post-mortem records is once the body is dead or the individual is dead. Antemortem records when the person was alive, his records. They try to compare the both the records together and they try to find out. This is done in generally from the medical department. How dentistry comes in action here, we also compare the anti and post-mortem records to find out the individual, whether this the X-ray or the radiograph or intraoral X-rays belong to same individual or not. Now, this was the conventional methods which has been followed since ages now. Now, technical advances have happened in the field of molecular biology. We are trying to find out the person by working at the genetic level, at a molecular level, and by analyzing the DNA. Now, the question is, do we have enough cellular elements in the tooth from where the DNA analysis can be done? Well, it is there, especially in the pulp region. We have the fibroblasts, odontoblasts, and the endothelial cells, and many more cells which can be taken, extracted from the pulp, analyzed for DNA. Which are the other sites and how this all this analysis can be done? We'll be talking about this in this presentation. Now, this is the different areas from where the DNA can be extracted. The most common way is the blood to be extracted and analyzed, or the simplest way is to take the hair follicle. So, if we see from the oral point of view or intro dental point of view, from the saliva it can be taken and from the tooth. If you look here, the easiest way would have been to take the blood sample from the blood or the hair follicle. Now, why we need the tooth for DNA extraction? The question arises whenever this application comes in picture, whenever a body comes to you which is totally decomposed, or the body is burnt, or there was a bomb blast, now you don't have any remnants remaining now. So here the DNA acts as a shield or an armor. If you know, the tooth has an enamel and dentine. Now this acts as a protection for the DNA inside. It does not allow the DNA to get withered away or decomposed. That's how where the DNA in the tooth is handy in doing the identification. Just to give an example how stable the DNA is in a tooth by extremes of the ages or extremes of the weather. This study, first one was done by Schwartz in the year 1991 at a temperature of 4 degrees centigrade. At extreme climatic variation, he could extract DNA after 6 weeks. Another example is a study by T.C. Bowles. What he found that even after 80 years, he could extract the DNA from the tooth. So this is the speciality of the DNA tooth here, that it acts as an arm or a shield where the DNA gets protected. Before we go into DNA analysis, I need for you to refresh the knowledge of the chemical structure of DNA. In order to understand the analysis part, you should know. I know you must have studied all this during your UG days or your PG days. It's just to refresh your knowledge. Now, DNA as such is a double helical strand. We call it as a polynucleotide change. Why polynucleotide? Because many nucleotides are there in this. Now, what is this nucleotide? Now, nucleotide is made up of a sugar, a base, and a phosphate. A sugar, a base, and a phosphate group. The sugar is a 5 carbon sugar. In case of DNA, it is deoxyribose sugar. That's we call as DNA, deoxyribose nucleic acid. In the case of RNA, it is a ribose sugar. That's why we call it ribose nucleic acid or RNA. Then we have the bases and the phosphates. Now, these bases can be of two types. You have the purines and the pyrimidines. Now, as a rule, always this adenine goes with thymine. That is, the blue one goes with the orange one. If you see, the blue one is always going with the orange one. Then we have the guanine and the cytosine. Then the green one always goes with the red one. Now, it is always because of this base pairing that the two helical strands are held together. Now, nowadays, easily we say the chemical structure of DNA, it's a double helical strand. It looks like a ladder which goes helically. Hello? Yeah, kindly mute your mics. There was some disturbance. Okay, now nowadays easily you can talk about the DNA structure. It's a helical strand. Okay, but this the scientists took around 100 years after the invention of the DNA to give this structure. All of us know the DNA structure was given by Watson Gray. In fact, they got Nobel Prize for this. But there are two more authors in this. The short story behind this. Now, this was considered as the scientific race of all time. Who will give the DNA structure? Even though the DNA was isolated, it took them around 100 years to give this DNA structure. Now, there were two groups of scientists who were working for this. We had the Watson and Crick and the Wilkins and Franklin. They were both working together as a team, Wilkins and Franklin. But there was some misunderstanding between the two, and Wilkins leaked all his their data of Franklin's to Watson and Crick. Now, ultimately, it was Watson and Crick who gave the DNA structure, and they got the Nobel Prize for this. So coming to the history, even though DNA was isolated in 1869, the DNA structure was given by Watson and Crick only in 1953. This is the first part. Then Jeffrey in the year 1985 talked about the DNA polymorphism. What do you mean by DNA polymorphism? Is that the pattern of DNA, the basis in the DNA is unique for each individual and gets transferred in a Mendelian pattern. This is called as DNA polymorphism. What is the potential application for this? It can be used for identify of the individual to know your parentage. For that also you can be used. Now, what are the techniques for DNA identifying the individual? One is by comparing the antemortem and the postmortem records, and other one is the DNA profiling or the DNA fingerprinting. Now, this is conventionally what we do. Compare the antemortem and the postmortem records. What we do here, we look for the tooth morphology. Any variation in the number is there. Maybe there is treatment done. Again, here, if you compare this with the antemortem record, there are two premolars with the root canal treatment done. The uniqueness in the lower dentition. This is the saucer-shaped restoration here, which can be seen over here. This is how we compare the antemortem and the postmortem records. Now the problem is that once you have the post-mortem record, you need to compare with all the records in that city. You have to need to collect all the records from the dentist. Nowadays we have software for this, for comparing, where all the data given by the dentist is put, and then you compare the antibiotic and the post-mortem record. So it becomes quite easier. Now, if you can compare the antemortem and the postmortem records, the what is the need of DNA fingerprinting? We have to why we have to go for DNA fingerprinting if just comparison was sufficient, but it has some discrepancies. Your comparison, the discrepancies are there, or the disadvantage is that the patient might have additional dental treatment done. Suppose the patient came to the dentist, he took a preoperative X-ray and he decides a treatment plan or root canal treatment. The patient undergoes a root canal treatment, but he has not taken X-ray after root canal. So the pre-operative X-ray is the only source of data with the dentist now. So once we what and post-mortem record, we have a tooth with an endodontic trait with gutta percha in this. So when you compare the records, it does not look the similar. That is one discrepancy can happen. The X-ray records, what we have got, it's not of good quality. Maybe there's a cone cord or any other problem is there. You cannot locate the records. Maybe the data which by the dentist was not given to the central agency. This could be one of the reasons, or the treatment was not done in the same area. Maybe he had come to India for a visit and he does not belong to India. He belongs to some other country. This could be one of the reasons. Or in India only he belongs to some other city. That could be another reason. These voids or the lacunae or the discrepancies form the basis of the DNA fingerprinting. Okay, now these are basics you should know about the genetics before knowing the analysis. Just to refresh your knowledge, what is chromosome? Now, DNA as such, if you see, it's a long strain which has multiple billions of copies of this thing, the basis over here. Now, chromosome is a condensed package of this DNA and it is found in the nucleus. Now, what is gene? That is the next thing you should know. What is gene? Now, as I told, in the nucleus we have the chromosome, which is a condensed package of DNA. Now, DNAs are long strands with millions of copies. Now, gene is just a segment of a DNA. It's just a part of a DNA which codes for a protein. The basic function of gene is to code for a protein, and it's also called as the coding DNA. This gene forms only 2 to 5% of the total DNA. Out of this total DNA, it is forms only 2 to 5%. The remaining 95% has no work. It has no work, and it's called as junk DNA, and or the non-coding DNA, because it does not code for anything. So now what is junk DNA? Till now what we talked about was chromosome, condensed package of DNA, gene, segment of DNA that codes for protein. Junk DNA, gene is considered as the coding DNA, junk DNA is the non-coding DNA. Gene forms around 2 to 5%, this is around 95%. Now this junk DNA, now this is what long strand, gene, or the coding DNA, remaining is the non-coding DNA. Now this non-coding DNA can be either interspersed or it can be repetitive. So here we have no two more terms. We have the tandem repeats, and how these tandem repeats, or especially the mini satellites, are unique. Now, this repetitive DNA sequence can be tandem repeats or can be dispersed. In this, we have satellites, mini satellites, or the microsatellites. This mini satellites or the short tandem repeats is unique for each individual. The pattern of this mini satellites that forms the basis of your DNA analysis. This is unique for each individual. If you find out the short tandem repeat for an individual, it will be unique for that individual. And if you do the analysis of the DNA from any of the body part, that and you find these repeats, you can identify the individual. So till now, what we are told, in the we have a cell. In the cell, we have nucleus. From there, we take out the chromosome. This chromosome has long strands of DNA. Take a segment of the DNA, that is gene, that is the coding DNA. Remaining non-coding DNA, there is no function, but it has the tandem repeats. Now these tandem repeats are unique for each individual, especially the mini satellites. These are called as short tandem repeats. It's like your ID card. It is unique for you. Then the serial numbers on this are unique for this your person. Look now, till before I talked about the source of DNA from the other parts. Now we are concentrating from the oral source, from where the DNA can be extracted. Well, it can be extracted from the teeth, from saliva, or from the biopsy material. Basically, teeth. What are the cellular elements we are getting? These ones: the pulp, dentin, and the cementum. From the saliva, we are getting the desquamated epithelial cells. The biopsy material can be either the epithelial and the connective tissue. Now, these are the different ways how the DNA can be extracted from teeth or saliva or the biopsy. Now, today, our area of concern is this teeth. So, these are different methods which can be taken. We'll be discussing about this in the following slides. First, what is the location of DNA in teeth? Where basically anywhere where you can get a cellular element, where the cells can be extracted, DNA can be extracted from there. The main source is your coronal and the radicular pulp. In this, what are the cells? Are they which are there? They are the fibroblasts, odontoblasts, endothelial cells, undifferentiated mesenchymal cells, which are found here. In addition to this, we have the lateral canals. From there also we can remove. Then we have the dentine here. In the dentine, we have the dentinal tubules with the odontoplastic processes. From there also the cells can be extracted. The less stable sites, your periodontal ligament over here, the cellular cementum over here. From where also the cells can be extracted for analysis. Now, what is the amount of DNA required and what is the available DNA? The amount of DNA which is required from the tooth is around 10 to 15 mg. I mean, which is available from the tooth is around 10 to 15 mg. It depends whether it's a molar or the incisors, so depending on the size of the pulse chamber. The required amount is in nanograms. So what is available actually, it is in milligrams, but the amount is which is required is in nanograms. Why we are then why this question come? Because sometimes the body, the dead decomposed body, what you have got is from which has been there buried for ages. I mean, 10 years to 15 years. Whether you can get those milligrams or nanograms, that this question comes. Now, what are the Variables affecting the for the amount of DNA in tooth, it depends on the type of the tooth. In case of molars, the pulse chamber is bigger. As already told, that amount of DNA we can be extracted will be more compared to the incisors, because the size of the pulse chamber is less. But it's not only the size of the pulse chamber which is important. It's the condition of the tooth is also important. Suppose the tooth is decayed and is totally non-vital. The molar is totally non-vital. From here, you cannot extract the DNA then, because the cellular elements are dead. Again, trauma. Because of the trauma, the tooth might have been exposed, the pulp might have been exposed, and the pulpal elements would have got decayed. And the period of time from the extraction till the body has been removed for analysis, or the buried body has been removed. That is also important. The more the time, the chances of DNA survival becomes less. The age of the child or the individual is also important. Once a child, as the individual is of younger age or smaller age, the pulse chamber is bigger. But as the child becomes old, the pulse chamber becomes small due to the aging process. Accordingly, the amount of DNA or the cellular elements that can be extracted will be less. Well, now, what are the steps involved in this? One minute. The first step in this is the DNA isolation. Now, how do you do the DNA isolation? From the blood, you have to remove the DNA fragment. Okay, now you are talking about in general. From the blood, you need to extract the DNA fragment. Now, this DNA fragment, the DNA as such is quite long. It is long strand. You have to divide it into small pieces. We call that as fragmentation of the DNA. Now, then this is called as also as the DNA digestion. After that, it is put on the agarose gel for electrophoresis, so that these fragments gets distributed on the agarose gel. The next step is that this agarose gel is not stable. It is fragile. So it gets transferred on the nylon membrane. This is known as southern blotting. This is done by a process called southern blotting. Then the next step, the radioactive isotopes are added. Hello, can you see the slides properly? Excuse me. Hello.
Yes, sir.
Can you see the current slide steps involved in DNA fingerprinting?
Yes, sir. Yes, sir.
Okay, because I got some message that the slides are not visible. That's why I was asking.
Sir,
Okay, okay, fine, fine. Some there was some issue, but yeah, if it is clear, fine. Maybe some
It's
other
clear.
persons have some problem. So I will continue. So what I have done, told that from the blood, the DNA has to be removed. Then it's broken down in small fragments. Then these fragments are put on the in an electrolytic chamber, so that these fragments can be distributed on the agarose gel. Now the problem with agarose gel, it is not stable, so then we get taken on the nylon membrane by a process called as southern blotting. Now in the next step, what we are doing, you are adding the probes. Now these probes gets attached to the short random repeats, so that we can look for the uniqueness. Now these short random repeats cannot be seen unless an X-ray can be is taken. This process is known as autoradiography. On once you take the X-ray, on that the short random repeats can be exhibited. This was in general how the steps are taken. Now, how can you do from the teeth? From the teeth, first step is to remove the pulp. From the pulp, the DNA needs to be isolated. Now the next step, that DNA is broken down into small fragments like DNA. Now only the first step is different. That is removing of the pulp, and from the pulp, the DNA isolated. Rest of the steps is almost the same. What we had told before only, that we have to digest the DNA, that we have to break down to small fragments. Then we have electrophoresis, southern blotting, and hybridization. Till hybridization, you cannot look for the uniqueness of the short tandem repeats, even though the probes are attached to it. For that last step, autoradiography has been to be done. Now we see each of these steps one by one. First, we look for the pulp removal. How the pulp can be removed? We can go for a vertical split or a horizontal split. From then, the pulp can be removed. Other way is that we can go for endodontic access, how we do for root canal treatment, and remove the pulp with the help of a broach. Now, these are the more crude methods, that is crushing the teeth, that could be done using motor pressure or grinding machines. The best way I would suggest would go for a endodontic access. Once you have extracted the pulp, from this the inorganic portion all is removed by these two agents. So what we are left is the organic portion, only the DNA is there. The next step is your analysis. For this, I told these are long strands with millions of copies of DNA. You need to break down or cut down these DNA into small pieces. This step is known as DNA digestion. For this, specialized molecular scissors like are used. Example is the enzyme ECOR. This enzyme breaks down the DNA into small fragments. The next step is the electrophoresis. For this, agarose gel is used. In this, wells are made. In this, the DNA fragments are placed, and then the current is passed. Now, DNA is negatively charged. So, as the current is passed, it starts moving towards the positive end. The larger fragments are not able to move that much. They stay towards the negative side, and the shorter fragments move more farther towards the positive side. Then the whole DNA is at the end of electrolytic cycle gets spread like this. Now on this you have the short tandem repeats also interpret somewhere. How do you look for this? For this you need to add the probes that we'll see later. So next step, now till now what we have done from the sample, we have extracted the DNA. Then the DNA we went for electrophoresis on an agarose gel, and the DNA gets separated by size. Okay, so far we have done only electrophoresis. Now this DNA is on an agarose gel which is not stable. It's transferred on a more stable medium, that is called as the nylon membrane by a process called as southern blotting. Now, once we have transferred it to a southern blotting, which is a more stable membrane, now we have the different DNA fragments on that. You have to look for the short tandem repeats. For that, how can we identify with the naked eye? For these labeled probes are added. This labeled probes gets attached only to the short tandem repeats. Now, even this process of adding the short label probes so that it gets attached to the STR, the short tandem repeats, is called as hybridization. Now, once this is done, uh, we cannot see still. For that, X-ray has to be taken. That is the next step. The next step is that now we have the gel with probes. Now that is already we have done hybridization. Now we have to put an X-ray film against it, and an X-ray is taken. Now this step is called as autoradiography. At the end of autoradiography, we get the X-ray of the person, which is unique for that individual, with the short tandem repeats and with the label probes, and this exhibits as grayish to black bands on the X-ray. Now the pattern of these bands is unique for each individual. That is the basis for your DNA fingerprinting. All said and done about the analysis. The one more thing what you should know is the PCR. Now corona time, we all of you know we said you need PCR test, PCR test. Now basically, what is this PCR test? Now, PCR stands for polymerase chain reaction. What is the role of PCR in DNA analysis? We'll see now. Before that, we should understand how the DNA replicates. Now, DNA is a double helical strand. Before the multiplication, it unwinds itself. From one strand, unwind in phase, and each of these strands acts as a template for formation of the next step. What is the need of PCR? What is the use of PCR? As I told, sometimes when you get the dead bodies, it has come out after a long time, maybe after 10 years. The amount of DNA that is extracted will be very less. And even though we need only nanograms, but the amount which we get even lesser. So how to make it amplified? How to make it more? So here the PCR becomes handy. Now this PCR is like a molecular photocopying machines. You put one DNA, unwind it, and make it go for multiple cycles, so that one DNA first we get two DNA strands, second cycle you get fourth DNA strand, like this. If you continue, you get millions of copies of DNA. So that Sufficiently, you have sufficient material for DNA analysis. Now, how PCR is done? In this, you should understand this. Excuse me. The DNA has a double helical strand, and we say these strands run anti-parallel to each other. It has a 5 dash end or a 3 dash end. The 5 dash end is attached to the 3 dash end of the other molecule. So they say they run anti-parallel to each other. How does the PCR is done? First step is denaturation. In this, you heat the DNA so that they unwind. Now we have two strands of DNA separated. Then we add a primer here. If you see this, primer always attaches to the 3 dash end. And in this is called this step where you are adding the primer is called as annealing. First step was denaturation, where we are unwinding the strand. It is done at a relatively higher temperature. Next step is annealing, where you're adding the primer. Now, in this next step is the elongation process, where the primer starts forming the new strand. Now, this formation of new strand always goes from the 5 dash end to the 3 dash end. Here also from 5 dash end to the 3 dash end. So, at the end of one cycle, from one DNA molecule, we have got two DNA molecules. Like this, we continue for multiple cycles, you will have sufficient DNA that is required for DNA analysis. Again, to see the temperature at what it is done, denaturation is done at 94 degree. What has happened? At denaturation, from one DNA molecule, we have got two strands. Then next step is annealing. It is done at 54 degrees here. Here, what we have done, we have the primer is attached. This always attaches as the 3 dash ends here and here. Next step is in the annealing. Now it has started forming the new strand. Now in the extension, at the end of the extension process, four new DNA molecules will be formed from each strand. That is, from we started with one strand. At the end, we will have two strands of DNA. Now, just to show a video, I hope it works. To how this process starts. First step is denaturation at high temperature. The two strands get separated. Next is annealing. Annealing, the primer gets added. In extension, from in the each molecule, each strand we form two molecules. So first is denaturation, high temperature. Annealing, low temperature, where primer is added. Extension, from each DNA fragment, you are getting two fragments. So at the end of cycle, from one DNA molecule, you got two DNA molecules. This was all about your DNA analysis and PCR. Now, what are the potential applications of tooth DNA fingerprinting? So DNA, as I've already told, it could be easily taken from hair follicle also, or from the blood. But in instances where DNA is not available from any other part of the body, then we go for two DNA fragmenting. Example for this will be plane crash. If you remember, when the Pakistan ex-president Ziaul Haq died, in the plane crash, this application was used. In again here also we can use for this. If there is charred bodies, we can use. In case of WTC collapse also, it was used for identifying the individuals. So, all in the end, to conclude, I would say that DNA, the dentists are no less than like a James Bond. His number is 007, our number is 02032, corresponding to the number of teeth which are there in the mouth. So, summarize how it is done. First, from the blood or DNA is to be removed. The DNA is divided into small fragments by molecular scissors. Then all the electrophoresis is done. Once this electrophoresis is done, it's on an agarose gel. It's not stable. You then take it on a nylon membrane by southern blotting. After that, you add probes. These probes get attached to the Schwann tendon repeats, cannot be seen by naked eye, only by auto X-ray, that is auto radiography. You can see these Schwann tendon repeats, which are unique for each individual. So here I conclude with this picture. Now this sunrise over here, it shows the beginning of a new era in the field of DNA forensics, and this is the steps or we need to find and take towards to reach that era. We know these tides over shows the different obstacles which you can come in our way, but we are definite that we'll overcome all these obstacles in the coming years, so that DNA profiling becomes a normal standard. Meanwhile, the gold standard is still remains the comparison of the antemortem and the postmortem records. That's all for today. This is my bibliography, what all references I have taken. Thank you. With this, the presentation is over.
Okay, sir.
Uh, is the moderator around there? I'm done with the presentation. Is the moderator around there?
Sir. Yes, sir. Be available, sir. So you're done. Your presentation is done, right, sir?
Yes, yes, done.
Okay. So anyone else? Do you have any queries or any feedbacks or any suggestions to be or any questions to be asked? You can just have a word with Dr. Alok Dubey right now. Hope everyone is clear with your things and everything. So I'll be sharing the feedback form now. So I request you everyone to just copy that form and just post the Google review. And if you just post a positive review with 5 star, you will have an additional in future. You will have some extra benefits on it. And also this will also, as we are conducting it as a free webinar, right? So your positive reviews always help us to move forward and create such more interesting topics like this, and to create more number of webinars in future, so that you can have and attend this at hassle free and free of cost on it. So once again, thanks for your time, and thanks for joining this webinar. Hope you are doing. Hope you all be safe and secure. So in future we'll meet for the another webinar. Thank you. Have a great day.